to Beyond Shodh, a podcast celebrating the research, innovations, and startups in the field of science, technology, engineering, and mathematics. With every episode, your host Dr. Sonia Balayan will bring you an inspirational and exciting STEM story through the eyes of experts, scientists, innovators, and researchers. A very warm welcome to yet another exciting episode of Beyond Her Shodh, a series dedicated to pioneer women scientists celebrating their journey and contribution to STEM research. Today, we are very delighted to have with us India's well-recognized leading scientist with a strong personality and an excellent mentor, Professor Paramjit Khurana. Professor Khurana is currently working as a senior faculty at the Department of Plant Molecular Biology, University of Delhi South Campus. She is one of the few plant biologists who is known for her immense contribution towards harnessing the application of plant biotechnology and genomics for increasing stress tolerance in crops and sustainable agriculture, especially in wheat and mulberry genomics. Her group also contributed towards the sequencing of wheat and rice genome as a part of Indian team. She has received numerous awards and honors for her contribution. Further, she is an active member of various academic and scientific societies. Her group published over 200 scientific publications with several patents. So without further delay, let's start yet another inspirational journey with our special guest, Professor Paramjit Khurana. Keep listening to Beyond Shodh and stay motivated. I welcome you, ma'am, to Beyond Shodh, and thank you so much for accepting our invitation.
Thank you so much. It's a pleasure to talk to anyone who's interested in any sort of a thing. It's very kind of you to say these kind of sweet words. But it's been a journey, that's all.
Sure, ma'am. Ma'am, to start with, can you please tell us about the research areas and the vision of your laboratory?
Yeah, I think when we started out, I was more interested in wheat biotechnology because wheat is the major crop plant.
Yes.
We say rice, but internationally it is wheat which is eaten more than rice being a tropical crop. But I worked on both rice and wheat. And that was my challenge. At the time when we started, only transformation of rice was possible. Wheat was not possible. So I took that up as a challenge and we tried to develop methods for agrobacteria mediated transformation in wheat. When I say wheat, we are talking of bread wheat, which is Triticum mystigum. But there are other wheats also. There is Triticum durum, which is a tetraploid, and there is Triticum dicoccum, which is a diploid also. And we have, which has medicinal plant properties as well. So we worked on all three of them, established transformation protocols for all of them, which was quite heartening because internationally I was asked to organize a workshop at the international level so that people could learn this transformation technology. But due to logistics, I think that program never worked out. But ours was the first lab which successfully transformed all wheat varieties. And in durum, we were first to introduce in a trait of commercial interest, that was for cereal cyst nematode resistance, which is not possible even till today against any nematode. And for that, CMET had organized an international workshop only on cereal cyst nematode in Tunisia. In Turkey, it's not Tunisia, in Turkey. And I was specially invited to participate in that. It was a very close-net workshop. Only people working on cereal cyst nematodes were there. So that was a good form of recognition, I would say, of our work. That yes, they are the only ones who have developed transgenics for that.
And then well deserved at that time.
Yes, I would say so.
Wheat is a very difficult system.
Yes.
Being a polyploid, it is very difficult, and at that time the genome is also not known.
No, not at all.
So, very commendable work. So, considering the resources available and how you have devised all these techniques,
It has always been difficult. I mean, For cereal cyst nematode, we worked on the durum wheat. Then in astagham, for drought and salinity, we took a, it was a double-edged sword, I would say, because we had the transgenics with us, but we could never do the segregation analysis, wheat being a hexaploid. So segregation analysis was impossible. We could not grow the plants in the field. You would never have enough populations in the greenhouse. So not only for cereal cyst nematode, but in bread wheat, we developed the double haploid technology. First raised the haploids in wheat, in the bread wheat, and then introduced in the gene of interest for drought and salinity tolerance, and then diplodized it. So we introduced in only one gene, Hodium vulgare, from Hodium vulgare, HVA1, and we fixed the genotype, and now we have the 9th generation and 10th generation with us, and there is no segregation. Because even when you hybridize it, when you get a pure line, this is actually a genetically pure line, because all the gene, the entire genetic complement is diplodized. So it is stable, it doesn't segregate. But unfortunately, because of a biotech policies and others, we could never take these to the field. But we've used them to study what happens in a genome, like a hexaploid, wherein you're introducing just one foreign gene. So what is the transcriptomic level changes? How does the plant reorganize? What is the effect of that, which has not been studied? Of course, the first paper was in PMB. The second we have published recently in Cells, which is a very high impact factor, but tells you how the transcriptome changes even when a single gene has been introduced. That too in a hexaploid, where you have multiple copies of the same gene also present. So that way, it's a good contribution, I would say, maybe from the basic science, because we had a good system. So I thought we should understand that what happens under different stress conditions.
And not just wheat, ma'am, another system that you also work on, Mulberry, which is also not a very common system.
No, that's a very challenging system.
Yes.
In fact,
so you have taken up all the challenging systems.
It's always been like that. I've been holding the bull by the horns only. It's a woody tree species. It's grown as a plantation crop. That means once you grow in the field, it's there for 7-8 years and so forth. But during my PhD, I worked on leguminous trees. And then DBT had a mandate for sericulture. And then I was invited they wanted a regeneration protocol so that the desired genotypes could be multiplied. So I started it up as a minor project, and then I got so engrossed into the whole thing of how the silkworm is so specifically eats only the leaves of one variety, not the others. So that stayed with me. And then
it's very curious to know.
Yeah, we have different species of mulberry, The silkworm will eat only indica leaf. The fruit type, that is the lavigator type, it will not eat that fruit leaf. The lavigator is only fruit type. In northern regions, if you go, you have a serrata type. That's a tree species. And that has traits for cold tolerance and termite tolerance. So I was more interested with these traits, and that is how we got to do the transcriptome of these things. And then when we were part of the rice genome work, at that time, after we published the rice genome in 2006 or so, because that was an international effort. So we always wanted to do something single-handedly. India mein humne kya kiya? So we did the chloroplast genome of mulberry at that time. A colleague mentioned, why don't we do that? I said, sure, that would be a good idea. So that happened to be the first complete genome from India. And we published chloroplast genome initially. Since then, I've been trying to do the nuclear genome of mulberry. And mulberry is also polyploid, like wheat. It's a complicated genome because it is openly pollinated and you get lots of things. But it's only last year that we've been able to do the complete genome of mulberry also. We've resequenced about 21 accessions too. So there's a complete database of indels, SNPs and others with mulberry as well.
And this will be a good resource for everyone.
Yes, there is an international consortia for sericulture where China, India, South Asian countries are there. And I'm always part of that meetings, those meetings.
Wonderful, wonderful.
All the transformation protocols, we have genes for cold tolerance, for drought, salinity, even for UV stress introduced in, and for biotic stressors also, bacteria, fungi and others in mulberry. And they've all been transferred to the major Central Sericulture Institute in Mysore, and they've been there for years. the Bangalore Academy, US University of Agricultural Sciences in Bangalore, they have also done the assay with the silkworm to see whether there is any side effect. That has also been done, published also. And this plant can be vegetatively propagated. So we thought it was a good thing to give it to the farmers, especially after the BT cotton was released. I thought this traits, mulberry also would be easily released because it is not consumed by humans, it is only consumed by the silkworm. Yet our policies did not allow that. So I've just handed over all the transgenics to those two places and see whenever is possible, because we thought mulberry being the plantation crop, it needs a lot of water. It's the second fastest growing plant. So you could also grow it, the transgenics, on wastelands where nothing else will grow. You could grow mulberry, and it's a plantation crop. Once grown, there is green cover maintained, and the farmers will have an additional source of income when there is a silkworm also. That was my ultimate goal, I would say. But I don't know when it would be realized, if ever it would be realized or not.
Recently the policies has, I think
Yes.
they were revisited and new policies are there.
Yeah,
so
now
hopefully
for the first
we will
time the
get-
crop plants have been allowed to do that. But what has happened is our transgenic lines are now they're allowing for genome edited ones.
Okay.
Ours is with classical techniques, but it doesn't matter. The brassica is also with classical techniques only, and that's a crop plant which is fit for human consumption. So if the government has moved ahead, maybe one day our transgenics will also see the light.
Fingers
of the day.
crossed.
Crossed, always hoping for the best.
Actually, this is just a news for everyone. Okay? This
Yeah.
variety is released.
Yeah.
But behind that, how much years of efforts,
Decades
of efforts
work.
were there? Nobody knows that.
Yeah.
So thank you so much, ma'am, for your contribution, and that too in very difficult systems.
Yes, and being in the university to do all that,
Yes.
in two very challenging systems. So people used to often joke and they used to say that you are always sailing on two boats, because I could independently talk of wheat, I could independently talk of mulberry, and I could independently talk of the genomics also,
Yes.
and the other things too. That's the way life has been. It's been a good
Wonderful,
journey.
ma'am.
It's been a good, satisfying journey. Yes.
So, ma'am, you have achieved so much in your research and academic career. So, what do you think your upbringing and the family support or the values that you have, absorbed from your family and childhood, what exactly they contributed in shaping up of
It's
today's
only
Paramjit Khurana?
2 days back I was telling this, yes, I would definitely say that your family background makes a big difference. My father was an army personnel,
Okay.
and
And now that aura also reflected in your personality. I don't know
yeah,
about
a
it.
disciplined lifestyle
Yes.
maybe. Yeah, because he was a very disciplined person, and he was an engineer without any engineering degrees. He never went to IIT, IIMs, because I'm talking of pre-independence days. He was basically a middle school person, and he enrolled in the engineers and electronics EME Limited, and he did innovations at that time. He learned tricks there. And he would be called on different sites to restore the equipment, the heavy equipment, the tanks and others which had any fault. And at that time, Sonia, in 1965 or so, he invented a couple of instruments
Oh,
and he got awards.
wow.
I was the youngest in the family. I didn't realize that at that time because I was still in my school at that time. But he kept one invention for himself. So when he retired, he patented that. He got the national award from the Invention Promotion Board. Fakhruddin Ali Ahmed as president, gave him the industrial award. Dr. V.V. Giri gave him the Invention Promotion Award. So I think that was there in my childhood always,
And that,
ingrained.
yes.
And he would never distinguish between ki ladki hai ya ladka hai. I think he was very neutral, equality, he believed in it. And he made me, I worked on lathe machines and other heavy machines also during my summer break and others. So he said, no, you must know. He says in Russia, the ladies drive the buses, the trucks and all the heavy equipment, so why not? So I have had a very encouraging beginning support. He was very good. I had the toughest supervisor, toughest. No Saturdays, no Sundays, except Holi and Diwali, we had only 2 days off
In PhD.
in PhD.
And who is your
Professor S.C. Maheshwari,
Oh,
the person who
yes.
discovered haploids. He was a hard task master, but I chose to be with him because he was the best available in the department. I was very clear I didn't want to go abroad at that time, although my parents were willing to send me abroad. I was not very keen myself. Yeah. And then I was fortunate, my husband also supported me in my efforts. I've had good mentors. I've learned from different people because I believe it's not only a teacher you learn from, it could be any XYZ whom you meet. Someone or the other has some qualities in him, and if you can imbibe that person becomes your mentor. So I think it depends from whom you pick up, what you pick up, and that passion, if anyone nurtures that, because with a supervisor, I used to often tell him कि आप हमें international projects देते हो and we are working locally. He says doesn't matter, you work double the amount. So anything which we were lagging, we were taught to compensate that by hard work, putting it more hours, and I think that has stayed with me. If you lack something in life, there is a way to circumvent that and go ahead further. I think I've imbibed a lot.
So I'm related to this only, that you being the role model for our generation of scientists. So actually, who is your role model, as you are saying?
No, I would say lots of people, not just one person.
So any
See,
special
learning is
who's in
primarily from your home. It starts from there. And then your teacher, your supervisor, I would say, teacher I would say. Because whom you choose as a supervisor is also your choice. You could choose a average teacher as your supervisor, you want the best, the most difficult one. You know, first of all, you would say कि मैं लेता नहीं अगर कोई हॉस्टल में नहीं रहता तो, because you would want 24/7 to be there in the lab. So we said, sir, काम मैं पूरा करूँगी. I'll never give you an excuse that I would not do this or that. You'll never have a complaint, but I will not join the hostel.
So you have not joined the hostel?
Not for my PhD. It was only sometimes when I had to do overnight experiments for a short time in my MPhil, when I had to do protein verification work, then I did for a couple of months. That's all. That too, because I knew my work demanded my presence. So I think it is your choices you make, whom you choose, and how rigorously you want to pursue your passion. That is what.
So is it your dream that you always wanted to become a scientist, or it just a destiny guided path?
No,
and you just kept on moving on?
Actually, I wanted to be a doctor.
Okay.
So right after my masters, I was hooked on to becoming a medical doctor. But at that time, we never had any entrance exam. So based on your percentages, and by 0.02%, I lost my seat. Had I applied the next year, I would have gotten.
Because
the
God
medical.
has the other plan for you.
No, at that time when I didn't get it, I said, मुझे doctor तो बनना है. So if I cannot be a medical doctor, let me pursue research. So I think in my graduation, maybe I was the only person who was very clear that I have to do a PhD.
And PhD, and then how you got interested in plant sciences?
That was by sheer chance. Actually, I was more interested in geology, which is very strange and surprising because we had no education. But someone said, कि नहीं, they make you visit all the natural parks and lakes and other things here, so you'll have a good time. And I was always a naturalist at heart. So I took up botany. And I really enjoyed doing that part also. Ecology or taxonomy, whatever it is, I've enjoyed that. So that is how I got into botany. It's a plant sciences.
Interesting. It's interesting to know that we want something else, and then we have something else, and then
That is true.
we become something else.
That is true. It has stayed with me. Even today, I mean, I enjoy the field trips very much. I enjoy the field knowledge also. Being a molecular biologist who's usually confined to the lab, but I try to have a balance and learn from nature.
And we also noticed that, ma'am, you have all the memories intact with you in the form of photographs and everything that you have collected over the years, and you have with us.
Yeah, it always.
Yes. Ma'am, what is the one milestone, or you can say the turning point that shaped the vision of your laboratory, that this incident happened and now we have so many leads and we have now going in this direction only? Is it some discovery
I
or
would not
something?
say a turning point, but yes, I had very dedicated students, because when I told them that they have to do wheat transformation, at that time we were not even sensitive to the fact that we are handling a hexaploid. We're not sensitive to it, wheat genome is not available. And couple of students actually tried to do that and they failed. Okay, but there were others who persistently tried and they were successful. I think that was the turning point when
you
they
are also saying that this type of technique was not done
earlier.
anywhere else.
Yes.
So you don't have any resource to look up to.
Yes.
So doing for.
But I think sometimes that is a blessing in disguise.
Yes,
because you know that you are the first one doing it. You are more innovative in your approaches. You're trying different approaches, and we tried a whole lot of approaches to do that. And couple of them, they were very successful. I would say Devashish Patnaik did very good. Duramwala, his transgenics were excellent. Then Harsh also did. He did the transcriptome work, of course. And then there are many others, like Neetu. She worked with Durans and others. I mean, and I gave her such a tough thing. She worked on entire vitamin E biosynthetic pathway, vitamin A pathway, two genes from both of them, and she got the transgenics. And since then, because in rice it is much easier. So students do that in rice. In wheat, it will take you not more double, maybe triple the time. But once you get them, that's it. Like Harsh was the one who did the double haploid. And before that, I think Devashish Patnaik was the first one who did the serial cyst nematode once. So I think these two things together were the turning point for us, that yes, we could do that.
And then how Mulberry came into picture?
Mulberry, I told you, once Dr. Tyagi after it.
Okay.
So I was invited, Dr. Tyagi was heading the sericulture, he was part of that committee. And there someone said that the biggest problem is we need to propagate the known varieties. And people don't do it because it's a woody tree species and regeneration protocols were not there. So he said, Paramjit, would you want to do that? I said, sure. Regeneration hi to karna. Then we did the transformation also. Unfortunately, I had very good technician at that. She was working as a technician. Later she did a PhD in Manaswini. She had gem of a hand. She did wonders. We had lots of students who then contributed. Manaswini contributed. Shalini contributed. She's a lecturer now somewhere in Bihar. But they did. So slowly then it, and when I saw that interaction, how the silkworm eats only the mulberry leaf and others, that is what intrigued me, that this is something special and no one else in the world, the West is not interested in it. You must pick up problems which are of local interest to you, to your country. And here I realized that this is something unique where it satisfies your intellect and you're able to contribute something. So it was true.
So ma'am, what all leads that you have got? And
in Mulberry, I mean, we are the ones who have established the regeneration, the transformation for all the varieties of mulberry, also the transcriptome analysis. There are 3 different types which we have done for drought, salinity, UV light resistance for bacteria, biotic stressors also. We have all of it, and now we have completed the nuclear genome also. Now we have added data, added information so that the sericulturists, the breeders can use it.
Okay. So ma'am, stress tolerance, as you talk about stress tolerance, especially your lab have done so much on heat stress tolerance in wheat. So is it possible that we can somehow have all stress tolerance, a climate ready crop?
Yeah, in one of my earlier interviews I said all weather.
Yes, I have read
somewhere. That was in India Today, I think. And that was because at that time we were very hooked up upon our mulberry transgenics that they can tolerate salinity, they can tolerate drought, they can tolerate heat stress, they can tolerate UV light, they can tolerate biotic stresses also. So we were at that time wanting to hybridize the abiotic stress tolerant ones with the biotic stress tolerant. We didn't do that because we knew we were not going, will never be able to take it to the farmer. But we had all the crops with us and we wanted to create one which was a rugged crop plant so that we could grow it in the wastelands. That was the aim, and the farmer would benefit out of that. I think it is possible. In wheat, we have done the heat stress, started it at the vegetative stages, at other stages, because the crop feels the heat at different stages. In southern India, it is at the tillering stage. In northern India, it's at the heading stage. And now at the stage where the spike also comes into play. So we have to address at different developmental stages how heat stress will be tolerated. We have more than 24 genes which we have shown proved as concept of principle that yes, they are contributing to heat stress tolerance. And all of them do it to a certain degree. It's like it's a big orchestra going on in the cell, the entire cell gets reprogrammed and you can pick up. And we are talking of only heat stress tolerance. We still haven't touched memory part of it. But yes, we have the epigenetics also role coming into play for heat stress tolerance. We have a couple of publications on that also. But still we've not done that for heat, but we have genes for that, and they can anytime be transformed for. We've shown that they confer heat stress tolerance in rice, which is all the more required because that is a tropical plant.
Well, usually also many plant researchers follow the same strategies, that we identify genes from transcriptome, epigenome, we have the candidate, then we show in transgenic system, yes, it is working fine. But taking this transgenic from lab to field, it's a big
gap. It's a big gap.
How can we just
I think you need different expertise for that, because one person cannot do everything here. Like number one, you have to choose your genes of interest judiciously, which gene to pick up. So when you have the genomic data with it, then you can pinpoint which allele you want to introduce. I'm talking of alleles because in wheat also all the RCAs will give you heat tolerance. The one which will give you for leaf, heading stage, for the grain stage and others. And if you introduce that, you know that this will happen. You can do that in wheat. Once you get it in rice or in other crop plant, whatever crop plant you're working on, then you need to tie up with the breeders because now you need to take it to the field level. And there, like in Mulberry, we've done that with the Agricultural Institute in Bangalore and the Sericulture Institute in Mysore. They are the ones who did the characterization. They are the ones who did the interaction with the silkworm and the transgenic mulberry too. But in wheat, we've not been able to collaborate with the plant breeders for non-scientific reasons, I would say, because it affects their economics. I was told, I don't know whether I should put that on record or not. I was told unofficially, don't ever say that we have wheat transgenics because it will affect your export. Same thing like for rice. Don't say you have basmati which you have transformed because it will affect your export value and so forth. But there are non-scientific, there are economic reasons and there are export policies, there are other policies which play a lot of role in actually deciding what will go to the field and whether the government is interested also in taking that to the field or not. That's a different thing. I realized it much earlier that wheat we will never be able to take to the field because of the policy reasons, not because we don't have it, we have it, but there are other secondary reasons for that. So I did not push for it, I would say. With Mulberry, I'm satisfied. At least I've done my job. Now it is for them to take it further. Let them do it. So we were planning to put in our application in our CGN, but I don't know.
Because most of the plant biologists are working on these mandates only. But how they got transformed in a product?
I think we did work a little too earlier. We did a decade earlier. Had we done the same work in this decade, this phase, maybe it would have been appreciated more. At that time, we were against a dead wall and there was a lot of anti-GM sentiment existing at that point of time. So I think our work, we were never successful. Not successful, I would say, because we never tried to take it to the field at that point. We were happy by by showing that as a proof of principle, this is possible, we can do this. Anyone who wants it, I was freely available and I was ready to interact with anyone. In fact, I had written to a couple of wheat breeders. I had a lot of interaction with DWR in Karnal. There's a Salinity Institute also there also. In Rajasthan also, in MP also, with the breeders. But people, I think the breeders were reluctant for obvious reasons.
Hopefully we got to see more such translational science.
Yeah,
I must say
I
that
think so.
we can change our research into products so that farmers also.
I'm sure in the coming decades there will be more youngsters like you would be doing that more. I am much more hopeful because the time is right now to venture into this.
So ma'am, coming to the representation of women in academics or STEM basically. So does the representation of women in top leadership roles somehow bothers you that we would be having more representation at the top leadership positions?
I think having a good scientist is more important.
Yes.
Whether it's in leadership role or non-leadership role, that really does not bother me that much, because I think when you're good at your subject, at your whatever you're doing, whether it is science, engineering or mathematics or whatever it is, I think you'll automatically be called into it. And not everyone vies for a leadership quality. I know I am happy working in my lab. I'm happy with that kind of a situation. But yes, if people, and now times are changing, people are putting women everywhere. The government policies are very positive. They want women representatives everywhere. That is a very positive step to take. But women taking on leadership roles, yes, there are many leadership women in leadership roles nowadays. It's much better. Women are much more, I would not say aggressive, but much more assertive than they were earlier. So good for them. Whosoever wants to. The time is conducive. The women need to express themselves, and you can come to leadership roles only after you've gone through others. If you are deserving, you will get it, definitely. It's not that the roles were not there; they were always there, and not only for women. I think even for men. You look at the number of men at the bottom and how many get into the leadership roles. The percentage wise, you have lots of. I shouldn't say. I don't know. I don't want to say anything, but majority it's a pyramid. So it's there for men. It's there for women also. Only thing is with women, yes, at the starting graduation level and others, they are not ambitious enough. They don't want to go up. They are happy with graduation, post graduation. So what is wrong with that also? You cannot.
There comes the sharp decline.
Yes, we say the decline.
Quite well.
Yeah, but if they are happy with teaching positions and others, that's good enough. I think that is also a very noble profession. If anyone wants to contribute, why not? You don't want everyone to be heading an institute or to be a researcher. So let everyone choose what they want to do. So I'm okay with that. I have no big hassles about it that women are not represented. Whosoever wants to be, they have enough opportunities, and if they want to be, they will be. And I see women coming up in a big way. So I think our country, our policies, our government policies are wonderful. They're much more liberal than the West. In West, they don't even get a maternity leave. Now see, you get 9 months maternity leave, 6 months maternity leave.
6 months.
In nowadays, it was just 3 months. And 3 months also, when I was in US, I gave birth to my son. I got only 15 days off I took, and that too unpaid leave. And I was back to work with no regrets. No, I raised up my kids also. But they laugh at you when they say कि 3 months की paid leave मिलती है, 6 months की मिलती है. But our government policies are very conducive. So, and people exploit that also. Let me, there's a little bit of pinch of salt. Let's be a little self-critical also. Where it is for your good. And now you get childcare leave also. It was unheard of. Have you ever heard any country give you childcare leave? So let's be thankful to our government also. Some of the policies are very good, very encouraging also, and that is the reason people want to come up in these government jobs and other places too, which is wonderful.
So ma'am, how did you balance your work, and because research is such a profession which requires so much of dedication, not 100%, I would say 200% dedication.
No, I call research is a 24/7
Yes.
job.
And then
You're never off.
especially after having kids, how did you manage both without any guilt?
No, it was very difficult. It was very, very difficult, I would say. It's not easy. But you have to strike the right balance. Every stage of your own development, you need to have your priorities right. They also need you. You need to make way for that. There was a time I would never go to international travels or never go out of the country, out of the city.
Okay.
I would say I'm not traveling. There was a phase I put a moratorium on myself because my family needed it. But after a while, when they were mature enough, then I would, judiciously make some way. Not that too much, but it's a very fine balance which you have to strike between what is required on the personal front and what is required from the professional front. And you can manage it if you know how to do it. I think it's just time management. I believe the highest official, your prime minister or president, they also have 24 hours a day only.
Yes.
Okay. Everyone has only 24 hours a day. And the amount of work they have to do, they do it, no? So it's time management, how they manage time. I think that is what Pandit Nehru also said. People used to say he used to sign and do work at night when others are sleeping. I have also had that habit of working at night when everyone is asleep, because you need to concentrate on something. And if I'm not able to concentrate at late night, I'm an early riser. So I used to take that time out for myself, work, because you are fresh at that time, and then attend to your other duties. So anyone in this world, whatever job you're doing, I would say 24 hours per day is the time you have. How you divide it is up to you, and that will depend on your priorities. What are your priorities and how you manage it? That's what discipline has taught me, that you have to prioritize it. You have to
So ma'am,
prioritize.
how you have created this ecosystem in your lab for women research scholar who are juggling between managing all these? What are your advice?
I've not done anything special for them. I've just been little more understanding with them. I've never said कि मुझे women ही लेनी है, men नहीं लेना. In fact, the boys in my lab I've been accused कि ma'am, you're too soft on the women. So I remember telling one of them, no, you don't know. They faced a lot of oddities, and he retaliated. He says, as if we don't have any trouble. And I think that was an eye-opener, because I learned from them also that even the boys doing PhD have their responsibilities.
They have different kind of responsibilities.
Yes. And it is not easy for them also. But with women, yes, there have been others. I mean, it's a very interesting case. One girl, I said, she was married, had a child, and she wanted to start a PhD. I said, not in my lab. You can go elsewhere, because when I given my 100%, I demand at least 90% from the students also. And if they're willing to give that, only then I take them. So her husband comes in, he says, ma'am, believe me, you will never have a time complaint from her. I will support her. So I said, okay. I took her because her husband was willing to support her. She completed her PhD. She had a second baby also during that same time period. And she did excellently well, exceedingly well, I would say. So that kind of a support is wanted. And if a girl, lady, married, unmarried, with child, without child, it really doesn't make a difference. If they are committed, like the girl I said, Manaswini, she was very good in transformation. She got married, she supported her family, she would do that, she took a longer time, but she was very good and she managed it very well. So every student I've seen who's done well, they've done well with looking after their other commitments also, all of them. And I think that is what I tell them, not to neglect anything else for your work. Work is important, but not at the cost of other things. So you need to be able to manage take care of everything together simultaneously. And I think most of my students have been able to do that. Most of them.
I have seen most of women in your lab at that time.
Yeah.
Yes. And all of them are very happy.
Even the boys, I think they've also taken up the responsibilities. So both ways.
So ma'am, what are your, you have trained such a task force of plant scientists. and everyone is doing their part. So what are your key mantras to everyone who is, just wanted to pursue her research career? So is there some specific?
No, usually I tell them that whosoever comes, you should be clear what you want in life. And whatever you're doing, do it with 100% dedication at that time. If you're doing a PhD, give it your full. Okay, if you're doing a competitive exam, you please do that, but give it your 100%.
Okay.
Don't do double kind of a thing. And sometimes there are students, they've left after doing 2 or 3 years also.
Yes.
I've had that kind of students also, but okay with them. They had other commitments. I have nothing against them. I only feel bad when I'm not taken into confidence. Okay, so that I know my project is going to end or not end, whether the person will take the project further or not. Okay, that's all. If I was taken into confidence, then I can look after the project more efficiently, I would say. I have nothing against others who don't do it, but when I am giving my 100%, I don't think it is wrong to expect 90% or 80% from my students also. And once they give that much, they also get addicted. They get used to doing hard work. So whatever they will do in life, they will always do well.
And I think they silently observe the guide, always.
There are many things which you imbibe subconsciously from your teacher. Today I realize what all I've learned from my supervisor, my teachers, my various teachers, I would say, not just one teacher. So yes, having good teachers is very important in life.
And after parents, teachers are the ones.
Yes.
Who will
They are the
be
ones who will tell you what to do in life.
They will scold you for your benefit only.
It's not only scolding, they're
Sometimes,
trying to
yes.
tell you what is the right way.
Yes, ma'am.
That's what I would say.
So yes, a good teacher is a blessing. And
you're lucky if you get a good teacher.
so ma'am, I have a
Such
little
as-
message for you from your student.
Warm greetings, dear ma'am and everyone. I am Dr. Vibha Gulyani Checker, Department of Botany, Girodimal College. Ma'am, I am thankful to you for your unblinking support and intellectual stimulation during my PhD. Ma'am, I cannot forget your ever-caring temperament. I still relish those meals you sent to me while I was about to finish my PhD. I cannot forget your care while I conceived my first child at that time, ma'am. Thank you so much for everything, ma'am. I could finish this journey of my thesis because of your unblinking support, ma'am. I still miss those chats with you. Ma'am, you hold a special place in my heart because even when I think of a thing that I am not able to do, I look up to you, ma'am. You could do each and every task with such a grace. You were a true researcher, ma'am, a true homemaker, and an ever-caring supervisor. Thank you so much, ma'am. Thank you so much from the core of my heart. I love you, ma'am.
Hello everyone. I take this opportunity to express my heartfelt gratitude to my PhD supervisor, Professor Paramjit Khurana ma'am, who has played a shaping influence on my academic research life. I am very thankful to ma'am for her unwavering support and his brilliance and keen guidance in this my PhD dissertation and research paper. Her scientific knowledge, expertise, patience and disciplined approach motivated us to perform and give our best. Her stellar careers, numerous awards and recognitions bears a shining testimony of her rich contribution to sciences. Working under her supervision has been a great learning experience for me. Words are not enough to thank you, ma'am, but thank you indeed, for you mean a world to me and for those who have known you and your work. Thank you so much, ma'am. Namaste.
No, it's a privilege, I think, to work with different students because every student is a separate personality. Every student has certain plus points, certain negative points. And I try to work on the plus points, ignore the negative points because, I mean, even I may be having many of them, but that's the way it goes. So I've been fortunate. I've had good team of students.
Ma'am, how
also.
many PhD students you had?
I don't remember now exactly, maybe 24, 25 or so of them. And many have done their dissertations, masters, and then left, and they're still in touch with me. So very proud to see them progress as well at different areas too.
In सबके behalf में ma'am, thank you so much.
Most welcome. Thank you. Thank you. जरूर बताइए.
What keeps you going even today with so much of passion, grit, and enthusiasm?
See, there was a time, Sonia, when my husband was my biggest support, and now when I want to give up science, sometimes I feel कि बहुत हो गया. Now it's my children who are supporting me. This is अरे आपने ये कैसे नहीं किया, आपको ये करना है. Now you must do this. Now you must do that. So I realize it's a role reversal which is happening now. You know, because somehow or the other they take a great pride that their mother's doing something and they appreciate it.
Yes.
Because I've always taken them with me, and now when I want to let go of little things, they don't let me let go. So they are my biggest source of strength, I would say.
Sure, ma'am.
So it's the world goes round, you know. So they tell me, no, it's time to do now other things. Now I want to not as an active researcher, but maybe take science to the society. Maybe do more society work. I'm more inclined towards that, so I enjoy doing that.
And ma'am, last part, but most important, your message to all the aspiring researchers.
Just hang in there. I would say, just hang in. Times will change. Keep doing your work. You will fail n number of times, but the next time when you fail, you'll fail better, and you will recover better, and you will find the way. That's all. That's all. I think it is just the consistency and hard work and believing in what you're doing, which is important. Nothing else.
Thank you so much, ma'am,
Thank you
for taking out time for us.
for this opportunity and for getting messages from all the students. We've had a good group. We've enjoyed. We've had a moments. We've had fights. We've had arguments also. But that's the fun part of science, doing science. I think you need to be receptive to the other viewpoint as well.
So ma'am,
Thank
we
you
wish
so
you
much.
all the good health and all the scientific discoveries with you keep doing this wonderful work for the research community. Thank you so much for taking out time.
Thank you.
Thank you so much.
Thank you.